All right, welcome back to the deep dive, where we take complex topics and give you the ultimate shortcut to being well informed. Today, we're plunging deep into the incredible engineering marvel that is your own body, the circulatory system.
Our mission for this deep dive is a clear one. We're extracting the foundational insights from a specific chapter on human physiology. We'll explore the key components of the circulatory system, from the blood itself to the heart's tireless pumping and the intricate dance of blood flow through your vessels.
Yeah, the goal is really to present each concept, mechanism, and definition clearly.
Exactly. Building a robust understanding without getting lost in an overwhelming detail.
That's right. Think of this as your essential guide to understanding of the vital machinery within you, focusing on what truly matters. We'll break down the chapter's core ideas so you walk away with a solid, graspable picture of this fundamental system. Okay, let's unpack this. To really understand how everything works, we have to start with the fluid of life itself, blood. It's a lot more than just a red liquid, isn't it? What's actually in it?
Absolutely. Blood is a dynamic mixture, really. A suspension of cellular components floating in a liquid called plasma.
Plasma.
And plasma is mostly water, over 90%, but those dissolved elements are crucial. The heaviest by weight are the plasma proteins, things like albumins, globulins, and fibrinogen.
Fibrinogen. That sounds important.
Oh, it is. Fibrinogen, for example, is absolutely essential for blood clotting. Actually, if you were to remove fibrinogen and other clotting proteins from plasma, what you'd have left is called serum.
Interesting. So plasma's complex, and then there are the cells floating within it. Where do all these different blood cells come from? Like, they don't just appear, right?
Right. They all start their life journey from a single type of undifferentiated cell, a multipotent hematopoietic stem cell found deep in your bone marrow.
Multipotent, meaning many possibilities.
Exactly. These are truly remarkable cells because they're multipotent, meaning they can mature into any type of blood cell. Your body needs red cells, various white cells following specific pathways of differentiation.
Okay, let's focus on the most common ones then. The erythrocytes, or red blood cells. Their main job is gas transport, right? Carrying oxygen around and taking carbon dioxide away.
That's their primary role, yes. These cells are just packed with hemoglobin, a special protein designed to reversibly bind with oxygen thanks to its iron atoms and also with carbon dioxide. It's a beautifully efficient system for gas exchange.
And their shape is really distinctive, that sort of flattened disc.
The biconcave disc shape is actually critical to their function. It gives them a very high surface area to volume ratio.
More surface means faster exchange.
Precisely. This large surface area is exactly what allows for rapid and efficient diffusion of oxygen and carbon dioxide in and out of the cell. It's a classic example of structure perfectly matching function.
So where does the body actually make these gas transporters?
They're produced in the red bone marrow. As erythrocyte precursors mature, they fill up with hemoglobin, and then, interestingly, they shed their nuclei and most other organelles.
Really? They ditch the nucleus?
Yeah, they become highly specialized just for gas transport. Young, still developing red blood cells, called reticulocytes, do retain a few ribosomes, which you can see with special stains.
And that tells you something.
It can. A higher number of reticulocytes in the blood can tell doctors your body is rapidly trying to produce more red blood cells, maybe after blood loss or at high altitude.
That brings up a good point. Hemoglobin needs iron. How does the body manage its iron supply? That must be critical.
It is. And the body is incredibly resourceful with iron. We store a significant amount in the liver bound to a protein called ferritin. Think of it like an iron bank buffering against deficiencies.
A buffer. Okay.
And when old red blood cells are broken down, mostly in the spleen, their iron is recycled. It's released into the plasma, binds to transferrin, an iron transport protein.
Like a delivery truck.
Exactly. Transferrin then delivers this iron right back to the bone marrow to make new red blood cells. This recycling system is incredibly efficient, moving about 20 times more iron daily than we actually absorb or excrete.
Wow. Okay, so what if the body needs more red blood cells? What's the signal to ramp up production?
The main direct control is a hormone called erythropoietin, which is primarily secreted by the kidneys.
The kidneys. Interesting link.
Yes, it acts directly on the bone marrow, stimulating those erythrocyte progenitor cells to multiply and mature into new red blood cells.
Now, speaking of red blood cells, we should probably touch on sickle cell disease.
Right. In severe cases, where someone inherits 2 copies of the mutated gene, sickle cell disease fully manifests. But individuals with just 1 copy, those with sickle cell trait, typically only show symptoms if oxygen levels drop unusually low.
So it depends on the dose, in a way.
Kind of. And what's truly fascinating is the evolutionary angle.
Mm.
These heterozygotes, the ones with the trait, are more resistant to malaria.
Ah, that explains why the gene persists.
Exactly. It's a really complex evolutionary trade-off.
Okay, moving beyond oxygen transport. Blood is also home to our immune system's frontline defenders. Leukocytes, the white blood cells, what are their main roles? They're not all the same, are they?
Not at all. Think of your leukocytes as specialized units in a highly coordinated defense force. They're all involved in immune defenses, but they have different tactics.
Like different types of soldiers?
Sort of. You have rapid responders like neutrophils, which are phagocytes. They essentially engulf and destroy microbes. Then there are cells like lymphocytes, which are the precision units protecting against specific pathogens by either producing antibodies or directly targeting infected cells. It's a very dynamic, adaptive army within you.
And what about those tiny fragments involved in stopping bleeding?
Ah, those are platelets. They're colorless, non-nucleated cell fragments, much smaller than red blood cells.
Fragments, not whole cells.
No, they're actually produced when large bone marrow cells, called megakaryocytes, literally pinch off bits of their cytoplasm into the circulation. Their primary job, as we'll get into later, is forming blood clots.
It sounds like the body has this incredibly sophisticated system for making just the right types and numbers of blood cells when needed. How is all that managed?
It's an incredibly complex system, yes. It's regulated by various hematopoietic growth factors, or HGFs. Erythropoietin is one we mentioned.
Right, for red cells.
Exactly. Others, like colony-stimulating factors or interleukins, target different white blood cell lines, and thrombopoietin stimulates platelet production. These factors don't just trigger production, they also prevent premature cell death. It's all about maintaining a very precise, dynamic balance for your body's needs.
Okay, so we have the blood components covered. Now let's zoom out to the entire cardiovascular system. The big picture, the heart, the vessels, and how blood actually flows through them.
Right. The system architecture. The circulatory system is elegantly structured into two main circuits. First, there's the systemic circulation. That's the high-pressure system.
Systemic meaning the whole body.
Pretty much. It starts from your heart's left ventricle, pumps oxygenated blood out through the aorta to all your peripheral organs and tissues, and then returns deoxygenated blood via veins to the right side of the heart.
Okay, systemic is out to the body and back. What's the other one?
That's the pulmonary circulation. This is the lower pressure circuit. It takes that deoxygenated blood from the right ventricle, sends it to the lungs via the pulmonary artery to pick up oxygen and drop off CO2.
Gas exchange central.
Exactly. And then it returns the now oxygenated blood via the pulmonary veins to the left side of the heart, ready to be pumped out into the systemic circulation again.
It's a figure eight, basically. How does blood actually move through all this? It can't just drift. There must be physics involved, right? Pressure.
Absolutely. It follows fundamental principles. The flow rate of blood, how much volume moves per unit time, is directly proportional to the pressure difference between 2 points in the system.
Higher pressure difference, faster flow. Makes sense.
Right. And it's inversely proportional to the resistance it encounters.
Like friction in the pipes.
Exactly. Think of it like water through a hose.
Huh?
Push harder, increase pressure difference, it flows faster.
Mm.
But if the hose is kinked or narrow, resistance, flow slows down.
So what creates this resistance in our blood vessels? What are the main factors?
Well, resistance is determined by three main things: the viscosity of the blood, the length of the vessel, and most importantly, the inside radius of the blood vessel.
The radius, the width.
Yes, the width.
Yeah.
And this is the crucial part. Resistance is inversely proportional to the radius raised to the fourth power.
The fourth power. Wow, that's huge.
It is. It means even a tiny change in vessel width has an incredibly powerful effect on blood flow. If a vessel radius is cut in half, the resistance to flow increases 16-fold.
16 times. Okay, so controlling vessel radius must be key.
It's absolutely the body's most important lever for controlling resistance day to day because vessel lengths don't change and blood viscosity stays relatively constant. So changing that vessel radius is how your body directs blood flow where it's needed most.
Let's turn our attention to the star of the show then. The heart itself, the central pump. What are its main structural features?
The heart wall, the myocardium, is primarily composed of specialized cardiac muscle. An interesting detail is that all internal surfaces, both the heart chambers and all blood vessels, are lined by this thin, smooth layer of endothelial cells, or endothelium.
Endothelium lining everything.
Pretty much. And the human heart, as you know, has four chambers: a right and left atrium, which are the receiving chambers, and a right and left ventricle, the powerful pumping chambers. They're separated by a muscular wall called the interventricular septum.
And it needs valves, right, to keep blood moving forward, not backward.
Absolutely essential. The heart has one-way valves. Between each atrium and ventricle are the atrioventricular AV valves, the tricuspid valve on the right side, and the bicuspid or mitral valve on the left.
Tricuspid, 3 flaps, bicuspid, 2.
Exactly. Then, at the exit of each ventricle, leading into the big arteries, are the semilunar valves, the pulmonary valve going to the lungs, and the aortic valve going to the rest of the body.
How do they open and close? Is it active?
No, it's passive. They open and close purely based on the pressure differences across them. When ventricular pressure is higher than atrial pressure, the AV valves snap shut. When it's higher than arterial pressure, the semilunar valves pop open. It ensures blood always flows forward.
What makes cardiac muscles so special compared to, say, the muscles in your arm? It works nonstop.
That's a key difference, its tireless nature. Unlike skeletal muscle, every single heart cell contracts with every single beat, about once per second, for your entire life without you thinking about it.
Incredible endurance.
It is. And it has very limited ability to replace its cells, so it has to be incredibly resilient. When a chamber contracts, picture a fist powerfully squeezing a fluid-filled balloon. That's the kind of force generated.
How does the heart muscle itself get the blood supply it needs to keep working so hard?
That's a critical point. The blood being pumped through the heart chambers doesn't actually nourish the heart muscle cells directly.
Oh, really?
Yeah. Instead, the heart has its own dedicated blood supply system. The coronary arteries. These branch right off the aorta just above the aortic valve and deliver oxygenated blood to the myocardium. Then the deoxygenated blood drains back via cardiac veins into the coronary sinus, which empties into the right atrium.
So it has its own plumbing. Okay, how does this tireless 4-chambered pump coordinate its contractions so perfectly? There must be an internal electrical system, right? A conductor.
There is, and it's quite remarkable. The heart has its own specialized conducting system. The key player, the natural pacemaker, is a tiny cluster of cells in the right atrium called the sinoatrial SA node.
The SA node, that's the one I've heard of.
That's the one. It spontaneously generates electrical impulses, action potentials that spread throughout the heart, setting the rhythm. It determines your heart rate.
So how does that electrical signal spread out to make sure all the chambers beat in the right order? Atria first, then ventricles.
Great question. The action potential, starting in the SA node, spreads rapidly across the atrial muscle cells, helped by connections called gap junctions. This makes both atria contract almost simultaneously.
Pushing blood down into the ventricle.
Exactly. Then the signal reaches the atrioventricular, AV node, located at the base of the right atrium near the septum. And here's a critical part. The signal propagation slows down significantly.
A delay. Why?
It's a crucial delay, about 0.1 seconds. This tiny pause is essential. It ensures the atria have completely finished contracting and the ventricles are fully topped up with blood before the powerful ventricular contraction begins. It optimizes filling.
Smart design. What happens after that pause at the AV node?
From the AV node, the signal accelerates again. It travels rapidly down the bundle of His, which runs within the interventricular septum, and then splits into the right and left bundle branches.
Heading towards the bottom of the heart.
Yes, these branches lead to specialized, very fast-conducting cells called Purkinje fibers. These fibers quickly distribute the action potential throughout the ventricular muscle mass, ensuring a near-simultaneous and highly coordinated contraction of both ventricles. Really efficient ejection.
So the SA node normally drives the whole rhythm, but what if it, or the AV node, starts to malfunction? Can something else take over?
Yes, other cells in the conducting system can generate their own rhythm, but they're normally overridden by the faster SA node. If the SA node fails, or the AV node blocks the signal, these other cells can become ectopic pacemakers.
Ectopic meaning out of place.
Sort of, yeah. They take over the rhythm, but usually at a much slower rate, maybe 25-40 beats per minute, which often isn't enough. An AV conduction disorder can mean the signal from atria to ventricles is slowed or completely blocked, causing them to beat out of sync.
Which would be bad.
Very bad. In severe cases, that's when an artificial pacemaker might be surgically implanted to restore a healthy, coordinated rhythm.
Can we actually see this electrical activity from outside the body? Is that what an ECG does?
Yes, precisely.
Yeah.
An electrocardiogram, or ECG, sometimes EKG. It's not recording the potential of a single cell, but rather the collective electrical currents generated in your body fluids by the simultaneous action potentials of many cardiac cells. Electrodes placed on the skin detect these tiny currents.
And those squiggly lines on the readout, what do the different bumps and waves mean?
A typical ECG shows three main characteristic deflections. First is the P wave. That corresponds to atrial depolarization, the electrical signal triggering atrial contraction.
P for atria.
Then comes the QRS complex. It's usually the largest deflection and looks more complex because it represents ventricular depolarization, the signal for the big ventricles to contract. The complexity reflects the varied pathways the signal takes through the thick ventricular walls.
QRS for ventricles contracting. Got it. What's the last one?
That's the T wave. It represents ventricular repolarization, the electrical signal for the ventricles to relax and reset for the next beat.
What about the atria relaxing?
Atrial repolarization actually happens around the same time as the QRS complex. So it's usually masked by the much larger ventricular signal and isn't visible as a separate wave.
This sounds like an incredibly useful diagnostic tool.
It truly is. ECGs are vital for diagnosing many heart conditions, rhythm problems, damage from heart attacks, AV block we mentioned, because defects in the heart muscle or conducting system will alter how those electrical signals spread, changing the shapes and timing of the P, QRS, and T waves.
But it only shows electrical activity, right? Not how well the muscle is actually pumping.
That's a key limitation. It tells you about the electrical events. Mechanical problems like a leaky valve might not show up on an ECG if the electrical activity is normal. You often need other tests for those.
Okay, so we have the electrical signal spreading. How does that electricity actually translate into the heart muscle contracting, squeezing the blood?
That process is called excitation-contraction coupling. In cardiac muscle, a key factor is calcium. The amount of calcium ions, Ca2+, that floods into the muscle cell cytoplasm during that electrical excitation is a major determinant of how strongly the muscle contracts.
More calcium, stronger squeeze?
Essentially, yes. Unlike skeletal muscle, the calcium released during a normal resting heartbeat isn't usually enough to fully saturate all the contractile machinery. There's some reserve capacity.
Meaning it can ramp up the force if needed.
Exactly. If more calcium comes in, say, due to sympathetic nerve stimulation during exercise, the force of contraction can be significantly increased. It allows your heart to adapt its power output.
That makes sense. But what prevents the heart from just locking up, like a muscle cramp that doesn't stop? A continuous contraction.
That's prevented by a critical safety feature, the refractory period of the heart. Cardiac muscle cells have a very long absolute refractory period.
Refractory meaning they can't be stimulated again right away.
Precisely. This period lasts almost as long as the muscle contraction itself, about 250 milliseconds. This long pause prevents the heart from being re-excited multiple times during an ongoing contraction. It makes summation of contractions and a sustained tetanic contraction impossible.
Which is good because
if your heart were to enter tetanus, a prolonged seized state, it wouldn't be able to relax and fill with blood between beats. It would immediately fail as a pump. That long refractory period is absolutely essential for survival.
Okay, so the SA node fires, the signal spreads, the muscle contracts thanks to calcium, but it can't stay contracted because of the refractory period. It has to relax to fill. This whole rhythmic sequence is the cardiac cycle, right?
Exactly. The cardiac cycle describes that recurring sequence of events, atrial and ventricular contractions and relaxations. We typically divide it into two main phases. Systole, which is the period of ventricular contraction and blood ejection.
Systole, squeeze.
Right. And diastole, which is the period of ventricular relaxation and blood filling.
Diastole, dilate and fill.
You got it. It's a beautifully coordinated dance of pressure changes, valve movements, and volume shifts, happening about 70 times a minute at rest.
Can we walk through the key events quickly? Let's use the left side of the heart again as our example.
Sure. Let's start in mid to late diastole. Both the left atrium and ventricle are relaxed. Blood is flowing passively from the pulmonary veins into the left atrium, and because atrial pressure is slightly higher than ventricular pressure, the mitral valve, the AV valve, is open, letting blood flow down into the ventricle. The aortic valve is closed because the pressure in the aorta is still higher than in the relaxing ventricle.
So passive filling first?
Yes. Then, near the end of diastole, the SA node fires, the atria depolarize, the P wave on the ECG, and they contract, giving a final little push of blood into the ventricle. This tops it off. The volume in the ventricle at the very end of this filling phase is called the end diastolic volume.
EDV volume at the end of filling. Okay, now systole.
Now systole begins. The ventricles start to contract, triggered by the QRS complex. As soon as ventricular pressure rises above atrial pressure, the mitral valve snaps shut.
First heart sound lub.
That's largely it, yes. But the ventricular pressure isn't yet high enough to overcome the pressure in the aorta, so the aortic valve remains closed too. For this brief moment, both the AV and semilunar valves are closed. The ventricle is contracting, but the volume isn't changing. This is isovolumetric ventricular contraction.
Isovolumetric, same volume. Okay.
Then, as the ventricle keeps squeezing, its pressure rapidly exceeds the aortic pressure. This forces the aortic valve open, and blood is powerfully ejected from the ventricle into the aorta. This is the ejection phase.
Whoosh, out it goes.
Right. The amount of blood left behind in the ventricle after this ejection is the end systolic volume, ESV. And the volume actually ejected, the difference between the starting volume and the ending volume is the stroke volume, SV. So SV, EDV, ESV.
Stroke volume. Makes sense.
Yeah.
What happens next?
The ventricles finish contracting and start to relax. Early diastole, corresponding to the T wave. Ventricular pressure falls rapidly. As soon as it drops below aortic pressure, the aortic valve snaps shut, preventing backflow from the aorta.
Second heart sound, dub.
Largely, yes. Now, for another brief period, the ventricular pressure is still higher than the relaxed atrial pressure, so the mitral valve also remains closed. Again, all valves are shut. The ventricle is relaxing, but the volume isn't changing. This is isovolumetric ventricular relaxation.
Another isovolumetric phase, okay.
Finally, as the ventricle continues to relax, its pressure falls below the atrial pressure, which has been filling with blood returning from the lungs. This pressure difference pushes the mitral valve open, and ventricular filling starts all over again, initially very rapidly.
And that rapid filling early on is important, you said.
Yes, because it means that even if the heart rate increases significantly, shortening the overall time for diastole, the ventricles can still get adequately filled early in the cycle. It ensures filling isn't seriously impaired, at least up to pretty high heart rates. Okay, that cycle makes sense. Now, the heart's overall performance, how much blood it actually pumps out over time, what's that measure called and how is it figured out?
That's the cardiac output, CO. It's simply the volume of blood each ventricle pumps per unit time, usually measured in liters per minute.
Liters per minute? How do we calculate it?
It's a straightforward calculation. You multiply the heart rate, HR, in beats per minute by the stroke volume, SV, the volume of blood ejected per beat, usually in milliliters per beat. So CO equals HR x SV.
Simple formula, powerful result.
Very much so. For a typical resting adult, maybe HR is 72 beatsmin and SV is 70 mLop. That gives a cardiac output of about 5 liters per minute. Think about that. Almost your entire blood volume gets circulated once every minute.
Wow. And during exercise?
It can increase dramatically up to 20, 25, even 35 liters per minute in highly trained athletes.
That's a huge increase. What controls our heart rate to allow for that kind of change? Primarily, it's your autonomic nervous system acting on the SA node. Sympathetic stimulation, your fight-or-flight response, releasing norepinephrine, speeds up the SA node's firing rate, increasing heart rate.
Makes it beat faster.
Right. Conversely, parasympathetic input, via the vagus nerve releasing acetylcholine, your rest and digest system, slows down the SA node, decreasing heart rate. These two systems provide constant fine-tuning.
Oh, okay, that's heart rate. What about the other part of the equation, stroke volume? What determines how much blood gets ejected with each beat? That can change too, right?
Absolutely. Stroke volume is determined by three main factors. First, there's an intrinsic property called the Frank-Starling mechanism.
Frank Starling. Sounds like researchers' names.
It is. It basically describes how the heart muscle responds to stretch. The more the ventricle fills with blood during diastole, meaning a higher end diastolic volume, or preload, the more forcefully it contracts during the subsequent systole.
So more stretch, stronger contraction.
Exactly. It's like stretching a rubber band further makes it snap back harder. This is a crucial mechanism for ensuring that, over time, the left and right ventricles pump out equal volumes of blood. It helps balance the two circuits.
Okay, Frank Starling's factor 1. What's next?
Second is sympathetic regulation, which directly affects the heart muscle's contractility. Contractility refers to the strength of contraction at any given end diastolic volume.
So separate from the stretch effect.
Yes. Sympathetic nerves releasing norepinephrine and epinephrine from the adrenal glands act on the ventricular muscle to increase its intrinsic contractile strength. This leads to a more complete ejection of the blood that's in the ventricle.
Pumping out a bigger fraction of the blood.
Precisely. We quantify this using the ejection fraction, EF, which is simply the stroke volume divided by the end diastolic volume, ES equals SVEDV. Increased contractility results in a higher ejection fraction. Sympathetic stimulation also makes the heart contract and relax faster, which is important at high heart rates to allow enough time for filling.
Makes sense. And the third factor affecting stroke volume.
The third factor is afterload. This is essentially the pressure the ventricle has to push against to eject blood into the arteries. Think of it as the load the heart muscle works against after the contraction starts.
So like high blood pressure in the aorta.
Exactly. An increased arterial pressure, increased afterload, makes it harder for the ventricle to eject blood, which tends to reduce stroke volume. In a healthy heart, other mechanisms usually compensate, but afterload becomes very significant in conditions like chronic high blood pressure or aortic valve stenosis.
Okay, EDV preload via Frank Starling, contractility via sympathetics, and afterload. Got it. Now, moving beyond the heart, the vascular system itself, the pipes, they play a huge role too, right? Especially in pressure and distribution. What's a key structural feature they all share?
A really important one. The entire inner surface of the circulatory system, heart chambers, arteries, arterioles, capillaries, venules, veins, is lined by that smooth, single-celled layer of endothelial cells, the endothelium.
We mentioned that lining the heart, so it lines everything.
Everything in contact with blood. These endothelial cells are incredibly active. They're not just a passive lining. They act as a selective barrier. They secrete substances that regulate vessel tone, clotting, inflammation. They're crucial players.
Okay. Now, the big arteries act like high-pressure conduits, smoothing out the pulses from the heart. But where does the real control of blood distribution happen? Which vessels are the main control points?
That would be the arterioles. These smaller arteries are the primary sites of resistance in the vascular system.
Resistance, meaning they slow the flow down.
Yes, and because they can change their diameter dramatically, they serve two major functions. They determine the total peripheral resistance, TPR, which is a key factor in blood pressure regulation, and they control the distribution of blood flow to individual organs and tissues based on their needs.
Like little adjustable valves directing blood where it's needed most.
That's a great analogy. As blood flows through the arterioles, there's a significant drop in pressure, from maybe 90 millimilli G down to around 35 millimilli G just before the capillaries. That pressure drop really underscores their resistance role.
How do they adjust their diameter? Muscle.
Exactly. Arterioles have a layer of smooth muscle in their walls. When this muscle relaxes, the vessel radius increases. That's vasodilation. When it contracts, the radius decreases vasoconstriction.
Vasodilation, wider pipe, less resistance, more flow. Vasoconstriction, narrower pipe, more resistance, less flow.
You got it. This allows for incredibly precise, independent adjustments of blood flow to different parts of the body. Arteriolar smooth muscle also has an intrinsic tone, a baseline level of partial contraction, which can then be increased or decreased by various signals.
What kind of signals tell these arterioles to dilate or constrict? Where do they come from?
They fall into two broad categories: local controls and extrinsic controls.
Local meaning right
Yeah.
there in the tissue.
Yes. Local controls are mechanisms within an organ or tissue that alter its own arteriolar resistance, independent of system-wide nerves or hormones. A prime example is active hyperemia.
Active hyperemia.
It means increased blood flow due to increased metabolic activity. Think about an exercising muscle. It uses more oxygen, produces more CO2, lactic acid, hydrogen ions, adenosine, potassium ions. These local chemical changes act directly on the nearby arterioles, causing them to vasodilate.
So the tissue calls for more blood itself when it's working hard.
Exactly. Another local control is flow autoregulation, where changes in the arterial pressure supplying an organ trigger compensatory changes in arteriolar resistance to keep blood flow relatively constant. This involves both those metabolic factors and direct myogenic responses, the smooth muscle itself contracting more strongly when it's stretched by higher pressure.
Okay, those are local signals. What about extrinsic controls from outside the organ?
The most important extrinsic controls involve nerves and hormones. The dominant nerve supply is from the sympathetic nervous system. Most arterioles are richly innervated by sympathetic neurons.
Fight or flight again.
Right. These neurons release norepinephrine, which binds to alpha-adrenergic receptors on the vascular smooth muscle, generally causing vasoconstriction. So increased sympathetic activity constricts most arterioles, increasing overall resistance and blood pressure.
What about the parasympathetic system? Does it control arterioles?
Generally, no. There's very little significant parasympathetic innervation of arterioles, except in a few specific locations like the salivary glands or genitals.
So mostly sympathetic for constriction. Any extrinsic signals for dilation?
Decreasing sympathetic activity below its basal level leads to vasodilation. Hormones can also play a role, like epinephrine, which can cause vasodilation in some beds, like skeletal muscle, via beta receptors, but constriction elsewhere via alpha receptors. And don't forget those endothelial cells, they release crucial local paracrine agents. One key one is nitric oxide, NO.
Nitric oxide, I've heard of that.
It's a potent vasodilator produced by endothelial cells in response to various stimuli, including blood flow, shear stress. It plays a big role in maintaining a baseline level of vasodilation and also helps inhibit platelet aggregation.
So lots of control mechanisms. Arterioles direct the traffic. Where does the actual business of exchange happen? Nutrients out, waste in.
That's the primary job of the capillaries. These are the smallest blood vessels, incredibly thin-walled tubes, basically just a single layer of endothelial cells resting on a basement membrane.
Just endothelium, no muscle.
No muscle, no connective tissue to speak of. This thinness is key for exchange. They form vast, dense networks, capillary beds that permeate almost every tissue in your body. Most cells are within a very short diffusion distance of a capillary.
Making exchange efficient.
Highly efficient. And another key feature, blood flow through capillaries is extremely slow. Even though each capillary is tiny, there are billions of them, so their total cross-sectional area is enormous, much larger than the arteries or veins. This slow flow maximizes the time available for substances to diffuse between the blood and the surrounding interstitial fluid.
Okay, slow flow, thin walls. How do substances actually cross those walls?
Three main mechanisms are involved. The most important for nutrients, oxygen, and metabolic waste products is diffusion.
Just moving down concentration gradients.
Exactly. Lipid-soluble substances like oxygen and CO2 can diffuse directly through the endothelial cell membranes. Water-soluble substances like ions, glucose, amino acids has to pass through water-filled channels. These are mainly the tiny gaps between adjacent endothelial cells, called intercellular clefts, or sometimes chains of fused vesicles forming temporary channels.
So small gaps for small water-soluble things. What about bigger molecules like proteins?
Plasma proteins generally can't diffuse easily across most capillaries, except in places like the liver. Some limited transport occurs via vesicle transport, endocytosis on one side, exocytosis on the other, but it's slow.
Okay, diffusion and some vesicle transport. What's the third mechanism?
The third is bulk flow. This isn't primarily about exchanging solutes, like nutrients or waste. It's about the movement of fluid, essentially protein-free plasma, across the capillary wall.
Fluid moving in or out. Why?
Its main function is distributing the extracellular fluid volume between the blood plasma compartment and the interstitial fluid compartment surrounding the cells. It helps maintain fluid balance.
What drives this bulk flow of fluid?
It's governed by a balance of pressures known as the Starling forces, named after the same physiologist as the heart law. There are 4 forces involved.
4. Okay, let's break them down.
2 forces tend to push fluid out of the capillary, filtration. The blood hydrostatic pressure inside the capillary, driven by blood pressure. And, if present, the osmotic force, due to proteins in the interstitial fluid, usually very low.
So blood pressure pushing out.
Right. And 2 forces tend to pull fluid into the capillary absorption. The hydrostatic pressure of the interstitial fluid outside the capillary, usually near 0 or slightly positive. And importantly, the osmotic force generated by the plasma proteins like albumin trapped inside the capillary blood. This is called the plasma colloid osmotic pressure or oncotic pressure.
Ah, the protein's pulling water back in.
Exactly, so it's a tug of war. Capillary blood pressure pushes fluid out, while plasma protein osmotic pressure pulls fluid back in. Normally, blood pressure is slightly higher at the arteriolar end of the capillary than the osmotic pressure, favoring filtration. As blood moves along, pressure drops, and by the venular end, the osmotic pressure might be slightly higher, favoring absorption.
So filtration out at one end, absorption back in at the other.
That's the classic view. In reality, in most capillary beds, there's a small net filtration across the entire length. A little bit more fluid filters out than is reabsorbed directly back into the blood.
Wait, if fluid is constantly filtering out, wouldn't tissues swell up?
They would, except for the lymphatic system. That excess filtered fluid, along with any leaked plasma proteins, enters the lymphatic capillaries and is eventually returned to the bloodstream. But if that balance is upset, say if plasma protein levels fall due to liver disease, reducing the osmotic pull back into capillaries, then fluid can accumulate in the interstitial space. That's edema.
Edema, swelling. Got it. Okay, so blood goes through the capillaries, exchange happens, fluid balance is managed. Now it needs to get back to the heart. What vessels handle the return journey?
Blood flows from the capillaries into venules, which are small veins, and these then merge to form progressively larger veins.
Veins. Are they just passive pipes back?
They are primarily low-resistance conduits for carrying blood back to the heart, but they have another very important characteristic. They are highly compliant, or distensible. Their walls are thinner and less muscular than arteries.
Meaning they can stretch easily.
Yes, much more easily than arteries. Because of this high compliance, veins act as a volume reservoir for the circulatory system. At rest, about 60–70% of your total blood volume is sitting in your veins. That's why they're often called capacitance vessels.
Holding most of the blood. Does this capacitance change?
It does. Veins do have smooth muscle in their walls, innervated by sympathetic nerves. When these nerves fire, the veins constrict. Because they're so compliant, even a small amount of constriction significantly reduces their volume capacity and squeezes that stored blood back towards the heart, increasing venous return.
So mobilizing that reserve volume. Now, how does blood actually get back to the heart, especially from way down in the legs, fighting gravity? The pressure must be pretty low in the veins.
It is low, so the return journey relies heavily on two assisting mechanisms, often called pumps, although they're not like the heart pump. First is the skeletal muscle pump.
Muscles squeezing the veins.
Exactly. When your leg muscles contract, like when you're walking, they compress the deep veins embedded within them. This compression pushes blood upwards, towards the heart. One-way valves within the veins are crucial. Here, they prevent the blood from flowing backwards towards the capillaries when the muscles relax.
Like check valves. Makes sense. What's the other pump?
The respiratory pump. It relates to the pressure changes in your chest cavity during breathing. When you inhale, your diaphragm contracts and moves down. This increases the pressure in your abdominal cavity, squeezing the abdominal veins. At the same time, the pressure in your thoracic cavity, chest, decreases.
Lower pressure in the chest.
Yes, this creates a pressure gradient between the abdominal veins, higher pressure, and the thoracic veins and right atrium, lower pressure, which effectively sucks or pulls blood upwards from the abdomen towards the heart. So just breathing helps venous return.
Clever. Muscle pump and respiratory pump. Plus the sympathetic constriction we mentioned. What about that fluid that leaks out of capillaries and doesn't get reabsorbed, the stuff that causes edema if it builds up?
That's handled by the lymphatic system. It's essentially a parallel drainage network. Tiny, blind-ended lymphatic capillaries permeate the tissues, collecting the excess interstitial fluid and any leaked proteins. This fluid is now called lymph.
Lymph. Okay, where does it go?
The lymphatic capillaries merge into larger lymphatic vessels, which eventually converge and empty the lymph back into the large veins near the heart, returning the fluid and protein to the circulation. Lymph flow is propelled by contractions of smooth muscle in the lymphatic vessel walls, the skeletal muscle pump, and the respiratory pump, similar to veins. And like veins, lymphatic vessels also have one-way valves.
So it's a crucial return system for fluid and protein. What if it gets blocked?
If the lymphatic system is damaged or blocked, say by infection, surgery, or parasites, fluid can't drain properly from the interstitial space, leading to severe swelling called lymphedema.
Okay, this whole system, heart, arteries, arterioles, capillaries, veins, lymphatics, sounds incredibly integrated and finely tuned. How does the body maintain overall arterial pressure? That seems critical for ensuring all organs get adequate blood flow.
Maintaining mean arterial pressure, MAP, within a certain range is absolutely crucial for survival. MAP is the average pressure driving blood flow through all your systemic organs.
And how is it controlled?
The fundamental equation to remember is MAP, cardiac output, COX, total peripheral resistance, TPR.
Pressure equals flow times resistance. Makes sense.
So all changes in mean arterial pressure must result from changes in either cardiac output, total peripheral resistance, or both. Over the long term, the single most important factor determining average blood pressure is your blood volume. And blood volume is primarily regulated by the kidneys through their control of sodium and water balance.
So kidneys are key for long-term pressure control via volume.
Exactly. It highlights the tight integration between the circulatory and urinary systems for maintaining homeostasis.
What about quick second adjustments in blood pressure, like when you stand up?
For that kind of immediate short-term regulation, the body relies heavily on the arterial baroreceptor reflex.
Baro meaning pressure. Pressure receptors, where are they?
They are specialized nerve endings, sensitive to stretch, located primarily in the walls of the two carotid sinuses, swellings in the carotid arteries in your neck that supply the brain, and in the arch of the aorta.
Strategic locations, what do they do?
They constantly monitor the pressure, both the mean pressure and the pulse pressure. The stretch. If arterial pressure decreases, the walls stretch less, and the firing rate of these baroreceptor nerves decreases. This signal goes to the cardiovascular control center in your brainstem.
And the brainstem responds how?
It orchestrates a reflex response to bring the pressure back up. It increases sympathetic outflow and decreases parasympathetic outflow.
Fight or flight kicks in.
Right. This leads to 1. Increased heart rate. 2. Increased ventricular contractility, stronger beat. 3. Widespread arteriolar constriction, increasing TPR. 4. Venous constriction, squeezing blood back to the heart, boosting CO via Frank Starling. All these effects work together synergistically to raise MAP back towards its normal set point.
So if I stand up too quickly and feel a bit lightheaded, that momentary dizziness is the pressure drop, and the quick recovery is the baroreceptor reflex ticking in.
That's exactly what's happening. When you stand up, gravity causes blood to pool in the compliant veins of your legs and abdomen. This reduces venous return to the heart.
Less filling, so lower EDV.
Right, which means lower stroke volume by the Frank Starling mechanism, leading to a temporary drop in cardiac output and thus arterial pressure. The baroreceptors detect this drop immediately, and the reflex kicks in, heart races a bit, vessels constrict to counteract it, and maintain blood flow to your brain.
And contracting your leg muscles helps too, right? The muscle pump.
Absolutely. Actively contracting your leg muscles helps squeeze that pooled venous blood back up towards the heart, reducing the initial drop in pressure and aiding the baroreflex.
Okay, let's consider another situation. Exercise. How does the cardiovascular system adapt to that huge increase in demand?
Exercise is a fantastic example of integrated cardiovascular control. As we said, cardiac output can increase dramatically, 5- or 6-fold. But just increasing CO isn't enough. The blood flow also needs to be redistributed.
Sent where it's needed most.
Exactly. During exercise, there's massive vasodilation in the arterioles, supplying the exercising skeletal muscles and the heart muscle itself. This is driven mainly by local metabolic factors, that active hyperemia we talked about.
So muscles get way more blood. What about other areas?
Blood flow is also usually increased to the skin to help dissipate the heat generated by metabolism. This often involves a decrease in sympathetic vasoconstrictor tone to skin arterioles. But simultaneously, sympathetic activity increases vasoconstriction in organs that are less active during exercise, like the kidneys and the gastrointestinal tract. Blood flow to the brain remains relatively constant.
So shunting blood away from the gut and towards the muscles and skin, what happens to total peripheral resistance overall?
Interestingly, despite the widespread sympathetic vasoconstriction in some areas, the massive vasodilation in the active muscles usually outweighs it. So total peripheral resistance, TPR, typically decreases during dynamic exercise like running.
Wait, if CO goes way up and TPR goes down, what happens to MAP? MAP equals COX TPR.
Good question. Mean arterial pressure usually increases slightly during strenuous exercise. The increase in cardiac output is proportionally larger than the decrease in total peripheral resistance. Pulse pressure, however, increases significantly because stroke volume increases and the blood is ejected more rapidly.
And all this depends on getting enough blood back to the heart, right? Venous return must increase massively too.
Critically important. The increased cardiac output during exercise absolutely depends on factors that promote venous return. The skeletal muscle pump working vigorously, the respiratory pump working harder due to deeper breathing, and the increased sympathetic tone causing venoconstriction, mobilizing that venous reservoir. It's a coordinated effort involving central commands from the brain, feedback from muscles, and resetting of the baroreceptors.
Amazing coordination. Let's touch briefly on some clinical situations where these mechanisms go wrong. High blood pressure or hypertension is common. What's usually the underlying issue there?
Hypertension is a major health problem, a big risk factor for heart attack, stroke, kidney disease. In most cases, about 90%, it's primary hypertension, meaning the exact cause isn't known. But what we typically see physiologically is an increased total peripheral resistance.
TPR. The arterioles are too constricted.
Generally, yes. Reduced arteriolar radius seems to be the main hemodynamic abnormality. Many factors likely contribute: genetics, obesity, high salt intake, stress, smoking, often leading to complex changes in kidney function, sympathetic activity, and vessel wall structure.
What about heart failure? What does that mean physiologically?
Heart failure means the heart simply cannot pump enough blood to meet the body's metabolic demands. It doesn't mean the heart has stopped, but that it's failing as an effective pump. This can happen for different reasons. Sometimes the problem is systolic dysfunction. The ventricle can't contract forcefully enough, maybe due to damage from a heart attack, leading to reduced stroke volume and ejection fraction.
Weak pump.
Right. Other times, it's diastolic dysfunction. Here, the ventricle muscle might be stiff, maybe due to chronic hypertension so it doesn't relax and fill properly during diastole. EDV is reduced, so stroke volume falls even if contractility is okay.
Stiff pump. Doesn't feel well. What happens then?
The body tries to compensate. The baroreceptor reflex gets activated by the low cardiac output, leading to increased heart rate and TPR, trying to maintain MAP. The kidneys retain salt and water to boost blood volume. But these compensations can become maladaptive, putting further strain on the already failing heart and leading to fluid congestion, like pulmonary edema, fluid in the lungs, if the left ventricle fails.
Vicious cycle. Another big one is coronary artery disease, CAD. That's about blood flow to the heart muscle itself, right?
Exactly. CAD means insufficient blood flow through the coronary arteries that supply the myocardium. The most common cause by far is atherosclerosis.
Atherosclerosis. Plaque buildup.
Yes, it's a disease process where plaques, accumulations of cholesterol, inflammatory cells, smooth muscle cells, connective tissue build up in the artery walls. These plaques narrow the vessel lumen, restricting blood flow, causing angina or chest pain during exertion, and they can also rupture.
Rupture, what happens then?
If a plaque ruptures, it often triggers the formation of a blood clot, coronary thrombosis, right at that site, which can completely block the artery. This cuts off blood supply to a portion of the heart muscle, causing the cells to die. That's a myocardial infarction or heart attack.
What are the big risk factors for atherosclerosis?
Major ones include smoking, high blood cholesterol, especially LDL, high blood pressure, diabetes, obesity, and a sedentary lifestyle. Genetics plays a role too.
And treatments?
Range from lifestyle changes and medications like statins to lower cholesterol, aspirin to reduce clotting, nitroglycerin to dilate vessels, to invasive procedures like angioplasty, using a balloon to open the narrowed artery, often placing a mesh tube called a stent to keep it open, or coronary artery bypass grafting, surgically grafting a new vessel to bypass the blockage.
Okay, finally, let's circle back to blood itself. How does the body actually stop bleeding when a vessel is damaged? The process of hemostasis.
Hemostasis, stopping blood loss. It's most effective in small vessels like arterioles, capillaries, and venules. It starts immediately with vascular spasm constriction of the damaged vessel. But the core processes involve the platelets and clotting factors we talked about earlier.
This is first.
Yes. First step is the formation of a platelet plug. When a vessel wall is damaged, underlying collagen fibers are exposed. Platelets circulating nearby stick to this collagen, largely via a plasma protein called von Willebrand factor, VGWF, which acts like glue.
So they stick to the damaged site.
Right. This adhesion triggers platelet activation. The platelets change shape, become spiky, and release various chemicals stored in their granules, things like ADP and serotonin. These chemicals act on other nearby platelets, causing them to become sticky and aggregate onto the initial layer.
A snowball effect.
Kind of. This platelet aggregation builds up a plug that quickly seals small breaks in the vessel wall. It's a positive feedback loop, but crucially, it's localized. Adjacent, undamaged endothelial cells release inhibitors like prostacyclin and nitric oxide, which strongly inhibit platelet aggregation, preventing the plug from spreading inappropriately.
Smart. So the platelet plug is step one. What's next?
The second major process, which reinforces the platelet plug, is blood coagulation or clotting. This transforms the blood in the vicinity of the injury from a liquid into a solid gel, the clot or thrombus. The main structural component of this clot is an insoluble protein called fibrin.
Fibrin. How is it formed? It's not normally floating around, right?
No, it's formed from a soluble precursor protein always present in plasma called fibrinogen. The conversion of fibrinogen to fibrin is the final key step, and it's catalyzed by an enzyme called thrombin.
Thrombin makes fibrinogen into fibrin. Where does thrombin come from?
Thrombin itself is generated from an inactive precursor in plasma called prothrombin. This activation requires a complex cascade of reactions involving numerous other plasma proteins called clotting factors, often designated by Roman numerals.
A cascade.
One triggers the next.
Exactly. There are two main pathways that can initiate this cascade, though they converge. The physiologically more important one for initiating clotting after tissue injury is the extrinsic pathway. It's triggered when damaged tissue cells expose or release a protein called tissue factor.
Tissue factor starts it off.
Yes. Tissue factor then activates factor X. Factor X, along with other factors like factor V, calcium ions, and platelet phospholipids, forms an enzyme complex that rapidly converts prothrombin to thrombin, and once you have thrombin,
it makes fibrin from fibrinogen, forming the clot meshwork. Precisely. Thrombin also strongly activates more platelets and accelerates earlier steps in the cascade, creating a burst of clot formation. The liver plays a crucial role here, synthesizing most of the clotting factors, including prothrombin, and it requires vitamin K for synthesizing several of them.
So vitamin K is essential for clotting. Now, what stops this cascade from running wild and clotting all our blood? And how do clots get removed once healing occurs?
Excellent points. The body has several anticoagulant mechanisms built in to control and localize clotting. For example, tissue factor pathway inhibitor, TFPI, secreted by endothelial cells, quickly limits the action of the extrinsic pathway initiator complex. Other plasma proteins, like antithrombin III, helped by heparin, and protein C, inactivate key clotting factors.
Checks and balances.
Definitely.
Yeah.
And once the clot has served its purpose and tissue repair is underway, there's a system to dissolve it, the fibrinolytic or thrombolytic system. This involves converting an inactive plasma protein called plasminogen into an active enzyme called plasmin.
Plasmin, like scissors for fibrin.
Exactly. Plasmin is a potent enzyme that digests fibrin threads, effectively breaking down the clot. Plasminogen gets activated to plasmin by various plasminogen activators, a key one being tissue plasminogen activator, TPA, which is released slowly by endothelial cells. Drugs based on TPA are used clinically to dissolve dangerous clots in heart attacks or strokes.
So, a system to build clots and a system to break them down, constantly in balance.
Precisely. This dynamic balance between clot formation, coagulation, and clot dissolution, fibrinolysis, is a beautiful illustration of the principle that most physiological functions are controlled by multiple regulatory systems, often working in opposition to maintain stability.
Ew. What an absolutely incredible journey through the intricate world of our circulatory system. I mean, from the single stem cells creating diverse blood components to the perfectly coordinated beating of the heart, the dynamic adjustments of blood vessels controlling flow, and the precise mechanisms to stop bleeding. It's just a constant vital symphony happening inside us every single second.
It really is. Understanding these foundational concepts, the components, the pump mechanics, the flow dynamics, the regulation, allows you to truly appreciate the sheer complexity, the resilience, and the adaptability of the human body. Each part plays such a crucial interconnected role, all working together tirelessly to maintain homeostasis and, well, support life itself.
And that's really what the deep dive is all about, trying to give you those aha moments, connecting the dots, and equipping you with knowledge that makes you feel genuinely well-informed about how things work. We hope this deep dive into the circulatory system based on this physiology chapter has given you a clearer, more engaging picture of these vital processes.
Keep asking questions. Keep being curious about the amazing machine that is you.
Thank you so much for joining us on the deep dive, and a very warm thank you from the entire Last Minute Lecture team.